All right, so for this slide here, this is slide 6. A gene is a coding unit, meaning a gene which is found on DNA molecule. Now, this is not saying that one gene is found on one strand of DNA on a DNA molecule. This is saying that a gene packet could be here and here and here and here, and these are all the exons, and then the intron spacers are spaced in between them. And RNA would go in and code for this particular gene sequence. In other words, it may code for number 1, may code then also for number 28 and 64 and 143. It would take all those gene splices out, put them all together, bring them over to the ribosome, and then create the protein. So where it says gene is a genetic sequence that codes for RNA, then in a protein coding genes, the RNA codes for a protein. So you would comment that DNA has several gene sequences that, when put together, will code for a specific protein. Okay. And let's go on to the next one here. All right, so on this slide, which is slide 7, that DNA is the genetic material of bacteria, viruses, plant and animal cells. Yes and no. Yes, it is found in bacteria, viruses, plant, and animal cells. No, it's not found in all viruses. There are some viruses that are RNA based, like the flu virus is RNA based. And this is why you got to get a new vaccination every single year, because the virus changes and mutates quite a bit. And so you got to update what your immune system can fight off. But plant and animal cells and bacteria all are DNA based. Okay. Now the next slide that you would have is this one. I changed the order up, and I was flipping a coin when I was first putting this out, and I left it alone, and then I went back and thought better of it, and put this slide then in front of it. So this is on your slideshow. This is your slide 9. This is your slide 8. I just reversed the order. You can do that by left clicking and moving slide 8. Slide 9 up to slide 8 by just clicking, holding it, moving it in between slide 7 and 8, and it will reverse the order. You don't have to. It's like, what came first, this or this? This is the background, but it doesn't tell you what it's the background for. This is the experiment, but doesn't give you the background. It's kind of like both of these need to be on the same slide, but there's too much information. So I left it in this way, and so I'll just leave it for this way for right now. This gets us back to Frederick Griffith's experiment. And if you went back to the timeline, which is back here, you can see Griffith was in 1928. And he talked about transformation. And transformation to him was transferring genetic information from one organism to another, whether it was parent to offspring, or in his case, one bacteria to another. Okay, and he was trying to prove that genes are actually found in DNA, not in the cell membrane, the protein of the cell membrane. So that's what his experiment was trying to prove here. So if I go back to this slide, what are all the mice for? Well, these little Mises, I did say Mises, I'll go back to old school cartoons. Was he a hater of mice? The answer is no, but he needed to be able to prove his point, and that was by injecting these mice with various strains of pneumococcus. Now, pneumococcus is a very nasty bacteria. Coccus, there's 3 kinds of bacteria. There's rod, spiral, and coccus. Rod looks like a big pill. Just think of it as like a capsule pill. Spiral, well, you can figure out what that is. It's just, it looks like a ribbon that's been curly-cued spiral. Coccus will look kind of like a brand new basketball. It will be really rough on the outside, but it'll be round. All right, so what's the difference between S strain and R strain pneumococcus? Well, first of all, what does the pneumo mean? Pneumo means lung. So this means that it's a bacteria that's round and it will affect the lungs. Okay, so what disease does it cause? It causes pneumonia. All right, so he's going to infect a mouse with pneumonia. And there's two kinds of strains. There's the R strain and S strain. Now, S strain is like you take that basketball that's nice and rough on the outside. It's brand new basketball. And you go over to the Nickelodeon studios. Why Nickelodeon? Because they are famous for one thing, slime. Take that basketball, dip it in a huge bucket of slime. It comes out all green and dripping, slimy, yucky, ooey gooey mess. And that's what S strain is it's ooey gooey on the outside. When introduced into a body, white blood cells will immediately try to attack it, but they can't because they get stuck in this slime and they can't penetrate to the shell of the outermost part of the bacteria, and so they can't penetrate it. So that little bacteria can basically do whatever it wants to do. In this case, it's going to irritate the linings of the host lungs and cause pneumonia, and the next thing they know, the mouse died. Well, unfortunately for the mouse, that was a good thing because that proves that his live S strain does cause pneumonia. And that's what he was trying to prove. So that's control. His next control is the R strain. So the R strain is rough. Now, he knew that if the mouse had a really good immune system, it will attack the pneumococcus bacteria and basically surround it, penetrate its outer coatings, and kill it. And the mouse should live. And the mouse did. Okay. They also knew back in the late 20s that if you heated up bacteria, it will kill it. And if it's dead, it should not cause pneumonia. And so he took some S strain that did cause pneumonia, heated it up, killed the bacteria, injected it into the mouse lived. So far he's got 3 controls. How many controls can you have in any one experiment? Don't know yet. We haven't found it. In infinity, it's basically you can have who knows how many controls, but you must have at least one. All right? You can have 1 million controls. It's a little excessive usually for an experiment, but theoretically you could have 1 million controls. All right? The next and last one here is his actual experiment. This is where he wants to show transference of DNA causes the R strain to act like an S strain. And the way he got this to do this is he heats up the S strain and kills it. Then he puts it in the solution with the R strain. Now the R strain's alive and it's hungry. What does hungry things do? They eat the other things, right? And so in this case, the R strain is going to eat the S strain. As it eats it, it basically takes in all of its contents. And because prokaryotes have what we call a naked DNA, it is not surrounded by a nuclear envelope or anything. It's over there in its own nucleoid region. The S strain's DNA is going to combine and anneal. It's called annealing. It's where it's going to hook into R strain's DNA and act just like normal DNA, and it will start coding. So that DNA now will code for everything the R strain used to have, and it will code for everything that the S strain had. And in doing so, the R strain will now turn into and start coding for a slime coat. Once it does that, it becomes a very virulent bacteria, and it will kill the mouse because the mouse cannot fight off any of this pneumonia. Okay, so the mouse will die. So here you would comment and put something to the effect that Griffith's experiment tried to prove transference of genetic material, so genes, from one organism to another. Okay, this is what he tried to prove. He did prove it. However, the people that were of this other camp over here that did not believe that DNA was where genes are found said, well, you did a great job at proving transference. We totally agree. He's like, whoa, what do you mean? If you agree that genes are found in the DNA, right? And they went, no, you totally proved this transference. And he goes, how so? When a bacteria eats another organism, it surrounds it, and when it does that, it takes that other organism's membrane apart. So it empties its contents into its own, space, and it digests those contents in there. Or in this case, it also takes its DNA and hooks it with its own. But in doing so, it also takes all of the membrane little pieces and puts in its own membrane. And they said, all you did was show that the proteins that were in the membrane are now in the membrane of the R strain. And so you showed transference of the proteins in the membrane. Congratulations, you proved our point. And he went, no, I didn't. And so it was a great experiment, but it did not prove where genes were actually found. And brings me to this slide. This slide is all of the background information that I just went over. So it talks about pneumococcus bacteria. It talks about virulence, which is a bacteria that causes some kind of disease. That's what virulence means and how bad it becomes. Capsular polysaccharide. This means it's like a capsule, so like a pill, that is made up of several different kinds of saccharides, which are different kinds of sugars. So polysaccharide is a variety of sugar. It's not a sweet sugar or anything. It's just like wood is made up of glucose, of molecules, but bonded totally different. And so it's no longer a sugar substance. That's why you don't see "ose" at the end of it. But the main thing here is that there are two groups, the smooth and rough pneumococcus, that cause pneumonia. And that the smooth is a pneumonia that causes the disease. Rough can cause the disease, but is easily fought off by the organism's own immune system. Okay, and I'm going to